Hello dear students, welcome in this channel Patel Science Academy. In this lecture you are going to know about the double fertilization. Double fertilization is the most important process of the angiospermic plants. What is double fertilization? The fertilization takes place twice. If fertilization is taking place twice or two times, it is called double fertilization. How it takes place, I'll explain in this lecture. One male gamete fuse with the female gamete. One male gamete, if it is going to fuse with the one female gamete, which is single haploid having half number of chromosome, this is also haploid having half number of chromosome. So this one male gamete, if it is going to fuse with one female gamete to produce the diploid cell, that is called zygote by the process of the first fertilization, it is called syngamy. Syngamy means the process of fusion of male gamete with one female gamete to produce the zygote. This is called syngamy or first fertilization. So after first fertilization, what happened? The second male gamete, if it is fused with the secondary nucleus, this second male gamete, actually there are two male gametes in the pollen tube of the pollen grain, out of which I have explained about the first one to produce the zygote, and second, what happens with this second male gamete? If this second male gamete is going to be fused with the diploid secondary nucleus as it is present in the embryo sac, there are 8 cell structure. This secondary nucleus at the center, there are two in number, that is why it is diploid. So this is haploid second male gamete, and these are the secondary nucleus. If fusion takes place in between the second male gamete and this secondary nucleus which is diploid, so there will be 3n production, because this is haploid single n and this is diploid 2n, so there will be 3n production structure, and it is called PEN, primary endospermic nucleus. So this is called triple fusion and second fertilization. So what happened with the first fertilization? By the syngamy process, the formation of the zygote by the fusion of the first male gamete with the female gamete. This is diploid formation, zygote. And what happened with the second male gamete? Second male gamete fused with the secondary nucleus to form the triple uh, N structure, PEN, primary endospermic nucleus. So this is called second fertilization. So two fertilization taking place here. As the result of first one, zygote is produced, which is diploid. As a result of second one, PEN, primary endospermic nucleus is produced, which is triploid. That's why this is called fertilization. Double fertilization is complex fertilization mechanism in flowering angiospermic plants, and it was discovered by the Navashin, by the scientist, and it is found in the liliaceous plants belonging to the Liliaceae family, like the lilium and the fritillaria in these plants. These are the example of this double fertilization plants. Let's see some more detail of this double fertilization. After pollen grain has reached the surface of the stigma, pollen grain starts germinating and forms the pollen tube. As you know, in my previous lecture I have explained about the pollen pistil interaction. The pollen grain enters on the stigma surface and it start producing pollen tube. What happens with that pollen tube? Pollen tube penetrates into the stigma, style and ovary chamber, and then enters the ovule. Then after the entry of the pollen grain, it start developing pollen tube. What happened with that pollen tube? It penetrate, or it enters into this style. As you know, this is style, this is ovary. So pollen tube enters into the style region, then from that style region it enters into the ovary region. From ovary region it goes into the ovules likewise. So the growth of the pollen tube is guided by the chemical secreted by the synergid cell. As you know, in the nucellus there are 8 cell structure, out of which the pollen grain, out of which the synergid cell is located toward the micropyle end. I have explained it in my one of the lecture. So what happened? Pollen tubes usually enters ovules through the micropyle. If pollen tube, there are three types of entry of the pollen tube into the nucellus. If it is going to enter into the ovule by the through the micropyle, through this region, as you know, this is micropyle region, this is chalaza region, and this is integument region. If pollen tube is entering into this ovule through this micropyle region, then this process is called porogamy. This structurally, diagrammatically, you can see here micropyle entering into this ovule through this pollen tube is entering into this structure ovule through this micropyle region, so it is called porogamy. Then what is this structure? Look at this structure here. Pollen tube is entering from this region, not from this region, so this is not porogamy. This is the region known as chalaza in front of micropiles. If it means pollen tube is entering into this ovule through this chalaza region. So pollen tube enters through the chalaza region. This process is called chalazogamy. So structurally you can see here this is chalazogamy because pollen tube is entering into this ovule through this chalaza. And third one is pollen tube is, if it is entering or piercing the integuments, so this is called mesogamy. So you can see diagrammatically here this pollen tube is entering into this ovule through this integument region because these layers are called integument layers. So if it is entering from the integument, then this is called mesogamy. So likewise there are types of three modes of entry of pollen tube into the ovule. First, porogamy, the entry of the pollen tube through the micropyle region. Second, chalazogamy, the entry of the pollen tube through the chalaza region. Third, mesogamy, the entry of the pollen tube through the integument region from this middle region through this covering. So these are three types. I hope you have understood it. The finally pollen tube enters or penetrates into the embryo sac of the ovule through its micropylar end, means by the porogamy. The pollen tube carrying male gamete penetrates into one of the synergids. As you know, synergid cells are, these are synergid cells. This is micropyle region, this is chalaza region, and at center there are two secondary nucleus. And this is egg. So besides this egg, the cell is called synergid cells. So what this point is indicating here, the pollen tube carrying the male gamete penetrates into the synergids. It goes for the synergids. Why it goes for the synergids? Because synergids content watery content is absorbed by this pollen tube. This pollen tube absorbs the watery content present here in the synergid cells. And why it is needed to the pollen tube? Because pollen tube rupture by absorbing that watery content and release the content. What are the content of this pollen tube? It includes two non-motile male gametes. So it means when this pollen tube enters into the nucellus through the porogamy, means by the micropyle region, it goes for the synergid, and synergid's watery content is absorbed by this pollen tube. Then this pollen tube and ruptures, and then this pollen tube ruptures and release its content, that is two male gametes which is coming from this pollen tube, from this pollen grain. As non-motile male gametes are carried through the hollow pollen tube, it is called sphenogamy. Because this pollen tube is hollow and non-motile pollen tube is coming through this pollen tube, that's why this is called sphenogamy. Sphenogamy means the entry of the process of carrying the male gamete through the hollow pollen tube. This is called sphenogamy, and that ensures the fertilization will takes place in future. Syngamy and the triple fusion means the formation of the diploid zygote and the formation of the triploid PEN primary endospermic nucleus are two events of the sexual reproduction in the angiospermic flowering plants. One more time, I'll explain you about syngamy. Syngamy, it is the fusion of the haploid male gamete with the haploid female gamete egg to produce the diploid zygote, as I have explained you diagrammatically. Triple fusion, the second haploid male gamete coming from that pollen tube fuse with the diploid secondary nucleus located at the center to form the PEN, producing the primary endosperm nucleus. PEN is formation. So this is the result of the second fertilization, and this is the result of first fertilization. Formation of zygote, the result of first fertilization. Formation of PEN, the result of second fertilization. PEN develops into the triploid endosperm. the the zygote develops into an embryo. What happens with that zygote? Zygote develops into an embryo. Syngamy is a type of the generative fertilization, and triple fusion is the type of the vegetative fertilization. Here, both the male gametes participates. Hence, these processes described as a double fertilization. So I hope till now you have understood double fertilization includes the first fusion of male gamete with the female gamete to form the diploid zygote, and the second fertilization, the fusion in between the second male gamete and the second male gamete and the secondary nucleus, that is for the formation of the PEN, primary endospermic nucleus, which is triploid 3N. So at last, let's see the significances of the double fertilization. What are the significance of the double fertilization? It is a unique feature of angiospermic. It is special feature, unique feature of angiospermic plant. It ensures that the parents plants invest a seeds with a food store, and only if the egg is fertilized, means only if egg is fertilized, formation of the seed will be there and it will be stored for the next generation. The diploid zygote develops into an embryo which develops into a new plant. The zygote is the next generation of the diploid zygote is the next generation of the any plant. So it ensures about the production of the next generation by that process. The triploid PEN develops into a nutritive endospermic tissue. PEN develops into endospermic tissue. I'll explain about this endospermic tissue and endosperm later in the next lecture. And it restores the diploid condition by the fusion of the haploid male gamete with the haploid female gamete through the syngamy, and it also helps to avoid the polyembryony. What is polyembryony? I have one more lecture about this polyembryony, so please subscribe my channel Patel Science Academy, and don't forget to share it and comment if you are having any problem. If you are any question in your mind, I will try